I had the hardest time figuring that out at first. I don't know why. And then I had to teach it, and it was like, oh,
That's how it goes. I mean, again, the three-dimensional process, trying to learn from three-dimensional, diagrams.
All right, good morning. In lab next week, you will become, in your imagination and mine, employees of Grizzly Pharmaceuticals. And the goal of Grizzly Pharmaceuticals is to wipe out Hanover fever. So we are going to contribute to that goal in our exercises over the next several weeks. Your assignment, if you will, over the next several days is to go into your lab manual, and hopefully you have it with you. And at the very beginning of the lab manual is what is referred to as the employee training manual. There's a lot of information in there that you need to digest before you start the lab next week. New terms, new concepts, new procedures that we'll be doing, all of which are very different from most of anything we've done so far this year. Explanations about as to what a plasmid is. And we gave you a little bit of an idea of that when we started the P-Globe lab. It's important to understand what a plasmid is. What is a restriction enzyme? Go into a little bit of that today. Process of electrophoresis, right? How we separate biological molecules based upon their migration through a medium under the influence of electrical charge. And how do we analyze that data? Right? There's a lot of new types of things we're going to be doing next week. The project that we have facing us over the next 4 weeks is, or can be, broken down into 4 parts, each of which represents the 4 labs over the next 4 weeks. Come on. Excuse me. Okay. Identifying, and this has partially been done, all right? Identify potential vaccine gene in the Hanover fever virus. Another division of Grizzly Pharmaceuticals has taken care of that. We will be attempting to clone that gene into a plasmid. Now, I want to caution you about this word clone. Some of you think of clones as something from Star Wars, or where you make a lot of identical sheep, or you have a pet cat and somebody makes a clone. That's not the kind of cloning we're talking about, although conceptually it's similar. Right? Cloning is the process of taking something and making lots and lots and lots and lots of copies of it. When IBM first came out with desktop computers, and then you had several other companies trying to do the same thing, those other companies were making what were called clones. Right? So anytime you make multiple copies of something, you can refer to as cloning. The cloning that we're going to be doing is making multiple copies of a particular gene. Okay? So don't confuse sort of the popular term for cloning and the way we're doing it. Right? We're going to clone the gene into a plasmid, take that plasmid, transform E. coli, much like we did with P-Glow, to produce what we hope will be transformed cells that contain the recombinant plasmid. This is a term that shows up in that employee manual. Be sure you understand that a recombinant plasmid is a plasmid that contains a piece of DNA from another organism. Right? We're going to then try to identify the bacterial transformants and find the one that contains the little piece of DNA, that gene, that can be used to make the vaccine.
Larry, can I say something real quick?
Sure.
The idea of this recombinant plasmid is an important one. You were working last week with this thing called the pea-glow plasmid. That does not occur in nature. What happened is that researchers took a bacterial plasmid and then were able to insert into it a gene for both green fluorescent protein and a gene for antibiotic resistance. So that pea-glow plasmid you've been working with is a recombinant plasmid. We're going to try to make our own recombinant plasmids in lab.
All right. The Hanover fever virus that we will seemingly be working with, or its DNA, is actually a bacterial virus called Lambda, right? But just to make it interesting, we're calling it something else. This Lambda virus, all right, Dr. Browder talked about it. It has a very interesting structure. It almost looks like a moon lander sort of a thing. We're going to be working with the DNA that's in the head of the virus, right? This DNA is just a little bit under 49,000 base pairs long and has genes for a variety of different parts of the virus. Any of the proteins, obviously, that make up the head or the tail or the neck have to be coded for, right? And so it's been mapped. We know where these genes are in the DNA. The gene that we're interested in is contained within this attachment, integration, excision segment in a piece of DNA that's generated when the landed DNA is digested with an enzyme called EcoR1 restriction endonuclease. Quite a mouthful, but the name will tell you a whole lot, and we'll go into a little bit more of that in a few minutes. Restriction endonucleases are probably the most important workhorse we have in trying to do molecular biology because it allows us to take a very large piece of DNA and cut it up into smaller, fairly well-defined, at least at the ends, pieces of DNA that we can then work with. Right? So as I mentioned to you, the Hanover virus vaccine gene that we're looking for is in one of the segments. It's 4,878 base pairs long. So hopefully we will be able to generate a transformed E. coli whose plasmid has contained in it, its recombinant plasmid, this piece of DNA. All right? So that's our challenge. All right, so primary goal to isolate the transform gene containing, transform E. coli containing that gene. The strategy, all right? Now, this describes the series of steps we're going to take over the next 4 weeks. We're going to take the Lambda virus DNA and the plasmid. The plasmid we're using is called PUC-18. PUC, I think, means University of California, but I'm not sure. All right, and as I mentioned to you in class, P simply is a designation as the plasmid. We're going to digest it with a restriction enzyme called EcoR1. Right, EcoR1 comes from E. coli. Right, we're going to take those pieces and we're going to attempt to attach the viral DNA pieces to the plasmid DNA pieces by a process we call called ligation. It's very, very similar to the ligation that Dr. Browder talked about when you replace an Okazaki fragment with DNA, and then you have to take those pieces of Okazaki fragments and connect them to the replicated DNA using DNA ligase. We will be using a DNA ligase to do this. We're going to take, so that's pretty much the first week. The second week, we're going to take our recombinant plasmids, transform them into E. coli. You will select one, an E. coli colony that is growing on plates that contain ampicillin, just like we did with P-Glow. You're going to grow it up, all right, and make lots and lots and lots of transformed E. coli that contains a recombinant plasmid. Then we're going to go in and we're going to isolate that recombinant plasmid from the E. coli. That's the third week. And then the final week, we're going to take those recombinant plasmids and see if any of us got one, or see which of the EcoR1 fragments you managed to clone. And ideally, hopefully, somebody in each class will have at least one clone that has the 4878 base pair fragment. As I mentioned to you, this is the kind of process that goes on in molecular biology labs all over the world, whether you're studying bacteria, plants, animals, protists, anything. All right? So this is a very straightforward and simple process that's used universally. All right? So we're going to start off on Tuesday by, I will provide you with lambda DNA. You can buy that really easy. Puck 18 we can buy. We're going to digest it with EcoR1 to make manageable pieces. The lambda DNA is really big, almost 50,000 base pairs long. It's too big for us to be able to insert it into a bacterium, right, using our transformation process. All right, so we're going to cut it up into pieces, mainly because we only want to study parts of it. We don't want to study the whole thing. We only want to study parts of it. So we cut it up so we can study parts of it. Right? So how do we cut the lambda DNA and the puck DNA into manageable pieces? Right? The answer is a restriction endonuclease. I'm not going to go into the origin of this term restriction. It has to do with selectivity of the viruses and bacteria that they infect. But endonuclease tells us something. What kind of a biological molecule do you think an endonuclease is? Yeah, it's an enzyme. It ends in ACE. All right? It's an enzyme that acts on nuclei, nucleic acids. And it's an enzyme that cuts nucleic acids inside the end of the nucleic acid. In other words, it doesn't cut off the A, T, G, or C that's on the end of the string of nucleic acid. It cuts somewhere internally, hence the prefix endo. Right? What do these little phrases have in common? Does anybody know? Abel was I, or I saw Elba, a Toyota. Madam, a man, a plan, a canal, Panama. We are not drawn onward to a new era or a nut for a jar of tuna.
They're palindromes?
They're all palindromes. Palindromes read the same way forward or back. Right? Abel was I, ere I saw Elba, a Toyota, and so on. The reason I bring this up is the places in a strip of DNA that the restriction endonuclease cuts is a palindrome. Right? Here we have a section of DNA, right, double-stranded, anti-parallel strands, and we have a segment where the sequence is GAATTC in this strand and GAATTC in the other strand. DNA sequences are always read in the 5' to 3' direction. So this is a palindrome. It reads the same way forward and backward. Okay? This happens to be the recognition site that our EcoR1 endonuclease cuts. Right? EcoR1 comes in here, but it doesn't cut it down the middle. This particular enzyme cuts it so that it always cuts between the G and the A. Right? And so when you do that, you create an overhang in each of the pieces. Right? An overhang called a sticky end. Now, other restriction endonucleases, ALU1, HAY3, BAMH1, HINDI3, EcoR1, each endonuclease, for the most part, has a unique sequence that it cuts. And this is a very useful tool. Right? Because when you take two pieces of DNA of different origins, the plasmid and the viral DNA, and cut them with the same restriction endonuclease, all the pieces have this same overhang, AATT. We're going to use that to recombine the fragments into plasmid. As I mentioned, when you have two types of DNA, let's say this is our viral DNA, this is our plasmid DNA, you cut with ECO, you get the same overhangs here, these sticky ends, so you can recombine them in such a way that you can take a piece of DNA from a totally different organism than your plasmid and insert it in the plasmid. Extraordinarily useful. Right? So, the overall picture of our plan, we'll have a plasmid, Puck18. We have a piece of DNA, lambda DNA. We're going to cut it with EcoR1, generating sticky ends in all the pieces. Right? We mix them together, throw in the DNA ligase. DNA ligase will hook the foreign piece of DNA into the plasmid, and we now have a recombinant that we can use to transform the chromosome, the bacteria. This is what we're going to do next week. The following week, we'll transform the bacteria and so on. All right? This does not happen, generally, it does not happen in nature. This is man taking nature and having it do things for us. It is biological technology. Right? The Puck18 or Puck19 plasmid, right, is fairly small. It's only a little less than 2,700 base pairs. It has an ampicillin resistance gene, the origin of replication. And there's a thing in there that we're going to make use of that's been cloned into it. It's called the LAC-Z gene. The LAC-Z gene codes for part of an enzyme that digests lactose. Lactose is a combination, it's a disaccharide of glucose and galactose. It's milk sugar, right? And so beta-galactosidase will break the covalent bond between glucose and galactose, right? It has been engineered to have a segment at one end called a polylinker. You don't need to remember that name, but it's a place where we can specifically put our chunk of DNA, right? Here's our plasmid, our LAC-Z gene, and there's an EcoR1 restriction site within that LAC-Z gene where when we digest the plasmid with EcoR1, creates an EcoR1 sticky, and so we can insert our piece of DNA into there. Now, the significance of that will become more obvious, not next week, but the week after that, because it helps us select for colonies that have a piece of lambda DNA inserted in it. All right? Oh, I don't want to go into that yet. So, be sure you read the introductory parts, the lab manual. It talks about plasmids, restriction endonucleases. We're not going to be doing electrophoresis. Well, yes, we will be doing electrophoresis this week, so be sure you read that segment. All right? Otherwise, you won't have any clue as to what's going on in lab. Because there's so much going on, I can't take a whole lot of time in the beginning to introduce it. All right? So be sure to read the material over. This is going to be a lot of fun because it's really different than stuff we've done before. You get to play with new tools in the lab, micropipetters and electrophoresis. Right? But come prepared.
Are you actually doing electrophoresis next week?
Yeah, we confirm the digestion of the plasmid and the DNA. And since we did that last year, take pictures with smartphones, part of what they have to hand in will be a description of what their digest looks like. I'm pushing it. A little bit harder. Eat you. Okay.
Okay, the stuff you're going to be doing in lab is going to be a direct application of the stuff we're talking about in this lecture sections for the rest of the semester. This is really applied molecular biology. This is biotechnology. And if I get on my soapbox for just a second, applications to research for producing products is really important. In this particular hypothetical scenario, you're involved in this process of cloning this gene, which is an important step in the development of a vaccine against a deadly viral disease. Okay? So products produced by biotechnology are incredibly important. However, I think that especially federally, the balance is tipped very much toward
funding of only applied research and not so much funding of basic research. Basic research is more kind of learning things to learn them, and they may have applications and they may not. Let me give you just a tiny little story. Way back in the 1960s, there was this old weird guy by the name of Werner Auber who was studying these bacteria that were infected by virus, and he discovered these weird enzymes that he called restriction enzymes. And he would go to conference after conference, year after year, talking about his wonderful restriction enzymes. And everybody would just go, who cares? Well, these ended up being the cornerstones of molecular biology. These enzymes are basically molecular scissors that allow us to very precisely cut DNA from two different sources, recombine it, and then that recombined DNA, if it's stuck into a plasmid, can be inserted into different organisms to produce all these wonderful different products. So applied research really would not exist were it not for basic research that's done that helps to discover a lot of the tools that applied research needs. We're going to be concentrating more on the application research now in lab, and you're going to be really practicing biotechnology. If you like doing this stuff, we offer an advanced biotech course that Dr. Morgan McCombs teaches. That's usually a junior-senior level course. And you get a lot more immersed into the biotechnology. And what's going to be really nice is that the opportunity is going to exist because we've got this new lab coming downtown of doing some collaborative projects with the folks at B2S Labs, and working in their facilities on research projects will be part of the mix of things. So this wonderful world of biotechnology is something you're going to get to experience at an elementary level now, and then if you choose at a more advanced level, and some of you may end up decide going into something in that kind of job arena. Okay? But you've got to understand some of the basics in order to get to that point. And so that's what we're doing right now, is we're trying to really get you to understand some of the really basics of molecular genetics, and particularly the dogma. So where we left off last time was we were talking about the part of the central dogma that had to do with translation. We had already talked about transcription, and remember that transcription is going to result in three primary products that have to come together in the proper way for translation to occur. What are those products? Transfer RNAs. Okay, so there are going to be genes in your DNA that are transcribed into these different transfer RNA molecules, about 45 different ones, and each one of those has to be charged up with its particular amino acid. That particular amino acid that's attached to the tRNA is really kind of defined by this anti-codon region down here, which is another crucial part of the tRNA. So we got all these charged tRNAs out here. Ribosomal RNAs are also transcribed. Remember, a big piece of ribosomal RNA is transcribed, and then it's chopped up into smaller pieces, and those smaller pieces associate with proteins, and then those form the ribosomal subunits, and then the ribosomal subunits eventually will come together if there's a message present as well. We'll step back. I didn't say this. Does anybody know where in your cells those ribosomal RNA subunits assemble? In a region of the nucleus, actually, called the nucleolus. Have you heard of that before? Okay. Sometimes with careful staining, if you look at cells, even under our microscopes, you can see this little dark stained area within the nucleus of the cell called the nucleolus. And that's where those ribosomal RNA subunits are put together. And then they have to be transported out of the nucleus into the cytoplasm, and then they come together to participate in this protein synthesis. Okay? So then at some point in time, a gene for a messenger RNA also had been transcribed. And then these things come together to form this translation complex. And then basically the tRNAs read the information encoded into the message. And in doing so, they bring the appropriate amino acids into position and join them together to form this polypeptide chain.
So as this ribosome moves down the message, one codon at a time, each time a new codon is exposed, then the appropriate tRNA that matches with it, codon, anticodon pairing, it'll have a particular amino acid that'll bring that into position and then grow that, add that to that growing chain. So by the time we reach the end of the message, we have synthesized this polypeptide chain. We now have a primary structure, and then that polypeptide chain will fold into a tertiary structure, and then that protein that's released will have some function in the cell. What dictates that function? Well, really, the information in the message. But what dictates the information in the message? The information encoded into the genes of the DNA. So again, our central dogma. DNA transcribed into RNA, RNA translated into protein. And what does protein do for a cell? Well, it makes its structure. Enzymes are proteins. Really, protein determines cellular phenotype. So genotype, your DNA information, determines phenotype, proteins produced, via this RNA intermediate. So DNA, RNA, protein. Okay? The ribosome has a structure that facilitates the interaction of all of these molecules. It has a binding site basically here that attaches to the messenger RNA, kind of a docking site, if you like. So that properly positions the messenger RNA. And it's got these 3 binding sites for tRNA. And really only 2 of those are occupied at any given time. The 3rd site is kind of the exit site. And Dr. Morton said, when I learned this, I remembered APE, APE, the A site, the P site, and the E site. Okay? So as the ribosome moves down the message, basically, always a new A site is exposed, and then what was in the A site is now shuffled over to the P site, and then what was in the P site now is ejected. So this allows these tRNAs to be recycled or reused in the process of translation. Because at one time, this was sitting in an A site, okay? And it had its particular amino acid associated with it, this one right here. And then it was shoved into the P site, and then eventually it was ejected, and then it can be recharged again if it hooks up with the proper charging enzyme, that amino acyl tRNA synthetase, okay? So pretty cool stuff. And this is kind of the basis of how your cells behave and what your cells look like and what they do because of the proteins that those cells produce. Okay? So during translation, the codons in the message are read by the tRNAs, and a polypeptide chain is produced. For understanding purposes, when we have something complex, sometimes our brains have a difficult time just wrapping our head around an entirely complex process. So one of the ways we tend to deal with that is to break things down into steps or phases. And that's what we're going to do with translation, is we're going to subdivide translation into kind of 3 events or 3 phases. There's initiation, there's elongation, and there's termination. You've already heard these 3 terms. Where have you heard these before? What process we've already talked about involves an initiation, an elongation, and a termination. Is it DNA replication? It's transcription, okay? When we talked about transcription, we talked about RNA polymerase attaching to a promoter. That's kind of the initiation event. That RNA polymerase copying the DNA template strand, that's the elongation. And then a termination event that results in that message or RNA molecule being released. As I said last time, one of the things that I want to really kind of hammer into you is that translation is a process that consumes a tremendous amount of energy. And the energy that's consumed during translation is provided by a molecule called GTP. GTP is guanosine triphosphate as opposed to adenosine triphosphate. It's kind of an ATP cousin. And in terms of your thinking, you can kind of equate GTP with ATP. The cell can actually convert easily GTP into ATP. You've actually seen GTP before. In the Krebs cycle, or the citric acid cycle, the molecules of energy that are produced by substrate level phosphorylation are actually GTP. And then the cell quickly converts those into ATP. I don't know what the significance of the fact is that translation relies on GTP rather than ATP, but it does. So we're going to keep track of this a little bit, not as much detail as we did in cellular respiration. But one of the things I really wanted to convince you is that this is an expensive process. And therefore, cells really don't want to spend energy unnecessarily making proteins they don't need. And understanding that's really important when you start to try to wrap your head around the idea of regulation of gene expression. The fact that cells can turn on and turn off either translation or transcription, and therefore save energy for the cell. Okay, so again, here's our kind of schematic model of what the process of translation would look like, kind of in midstream, okay? This much of the message down here has already been read, and then here is the next amino acid that's being added to this growing polypeptide chain. So we move down the message, codon by codon, adding amino acids to the polypeptide chain. Okay, so what's the first step? First step is initiation. The initiation codon that is part of the sequence of messenger RNA is the codon AUG. And remember that I told you that this is kind of weird in a little bit, in that this is not only a start codon, but that also specifies an amino acid. Remember the name of the amino acid? Amino acid called methionine. Okay? So technically, when proteins are produced, they all begin with methionine. Now, in reality, once a protein is produced, it's not uncommon for that first amino acid to be lopped off. So functionally, after the protein's made, it might not begin with methionine any longer. So we've got this initiation codon. So the tRNA that binds to this codon has the anti-codon UAC, and that particular transfer RNA is charged up with the amino acid methionine. This particular process requires energy. Now, I'm going to kind of dismiss this because this only happens once in the process of making a polypeptide chain. So this is kind of inconsequential use of energy. We're going to see more energy impacts later on. This also requires various proteins that are called initiation factors. And we're not going to delve into all those initiation factors. That's way beyond where we want to go. So, here's what happens functionally. We've got this charged initiator tRNA with its anti-codon UAC, and the enzyme, the amino-ACL tRNA synthetase, has loaded that up with the amino acid methionine. Okay? It's very important that that enzyme does that properly. Okay? Because basically in the genetic code, this code word here says methionine. Okay, so there's got to be the proper correspondence. When initiation begins, we temporarily form a very unstable complex. We've got this small ribosomal subunit
that positions on the message a little bit ahead of the start codon. In molecular biology parlance, this area ahead is called upstream because translation occurs in this direction, which is downstream. So this is kind of opposite upstream from the start codon. And there's a binding site on the small ribosomal subunit that recognizes this region and positions things properly. And then this tRNA comes in here, and it bonds by hydrogen bonds with its anticodon pairing with this codon. But that bonding is only hydrogen bonding. That's really weak. So this is a very unstable complex. But very quickly, GTP is hydrolyzed, and as a result of that, the large subunit then settles into place. And it settles into place in such a way so that this particular initiator tRNA is now positioned in the P site of the ribosome. Okay? So the A site is still empty. The P site is now occupied by this initiator tRNA carrying methionine, and the E site is empty. Okay? So that's initiation. It happens once. And then it's going to be preceded by many different elongation events. Every time a new code word is read, they'll go through an elongation cycle. We'll show you the elongation cycle in just a second. Okay? Okay. The next step is elongation. So basically, this is the series of events that happens over and over again as each additional code word in the message is read and interpreted. The elongation cycle is really fast. It takes about 60 milliseconds for this elongation cycle to occur. During the elongation process, one of the codons is read, and then when that's read, then we will move the ribosome down one more codon, and then another elongation cycle will read that. Okay, so interpret the codon, move the ribosome down, interpret another codon, move the ribosome down. Those are elongation events. Every time we bind a tRNA with its appropriate amino acid to that A site, that's going to chew up a GTP. So every time there's an elongation event, we consume a GTP. How many elongation events are there? As many as there are codons in the message. So for every codon you read, you chew up a GTP. That's getting expensive. Okay? And what does an elongation event result in? An additional amino acid being added onto the polypeptide chain. So basically, for every amino acid you add to the chain, it takes a GTP. So if I've got a protein that's 3,000 amino acids long, and that's a pretty short protein, that's going to chew up 3,000 GTPs. Okay? That's a big investment of energy. In between or during each elongation event, there's also a translocation phenomenon. That's when the ribosome moves down the message to read the next codon. And every time you translocate, that requires another GTP too. So energy demands are starting to add up pretty quickly. So here is the elongation cycle. So going from here to here is one elongation event. And every time you do this, basically a new codon is read. Okay, so you can see here, a GTP is required here, a GTP is required here. Okay. Now, unfortunately, this shows you kind of elongation in progress. Okay, so we've already had an initiation event, and then 3 elongation events that have occurred previously. So this part of the message has already been partially read. We've still got a lot of the message to read, okay? That's what the subsequent elongation cycles will do. So, we'll start here. This whole process, remember, takes about 60 milliseconds. The E site is empty. The P site contains a tRNA to which all of the previously joined together amino acids are attached. So everything's hooked now to this tRNA. Remember when tRNAs came in, they only had a single amino acid. Now they carry all of the amino acids that have previously been assembled together. The A site is unoccupied. So here is a tRNA that will come into the A site. It will only come in if its anticodon is complementary to the code word in the message. Okay? When that happens, when that position's into place, and I'm going to have to check this, I just saw this. They're implying here that it requires 2 GTP. I thought it was just 1. I'm going to have to check this out and see. But it does require GTP for this tRNA to come into the A site. Okay? So now we've got both sites occupied. The P site has all of the previously joined together amino acids. This has the incoming amino acid. A bond is now going to form between this amino acid and this polypeptide chain. What kind of bond is this? It's a peptide bond, okay? Peptide bond formation is catalyzed by an enzyme called peptidyl transferase. And peptidyl transferase is a ribosome. So one of these RNA molecules that makes up this big subunit is actually catalyzing the formation of this particular peptide bond. Okay? So now, everything is attached to this tRNA. And this bond is broken. So this tRNA right now no longer has anything attached to it. And now the growing polypeptide chain is all attached to this tRNA in the A site. So what happens? Both sites are occupied. So the ribosome slides down the message by 1 codon. It slides down this direction. So this is now in the E site. That's ejected. This now goes into the P site. And the A site will now be open because we've now hovered over a new code word in the message. We're moving this direction. So now we've completed an elongation cycle. So now we're ready to read this code word. Bring this this transfer RNA in. Hook these together. Move the ribosome down one more code word. This tRNA leaves. This one goes over to the E site. And now we're ready to accept a new tRNA. So it just kind of chugs like a train down the track, one code word at a time, and bringing in the amino acid that is carried by the tRNA that is specified by the code words in the message. Okay? So depending upon which is correct, for every elongation event, for every amino acid added, we have to consume at least 1. I thought just 1 more here for a total of 2, but maybe as many as 3 GTPs. So we're really chewing up a lot of energy. And remember, to get this amino acid attached to the tRNA, that requires an ATP as well. Okay, termination. Remember that there are stop codons in the genetic code. There are 3 of them. And those stop codons, there's not a tRNA that will bind to them. Instead,
proteins that are called release factors bind to those stop codons instead. So, this is termination, okay? We've translocated now, and this tRNA carries all the previous amino acids that have been hooked together during elongation events. Now the A site is exposed over this stop codon, but there's no tRNA that matches with those. But instead, there are release factors that bind in that A binding site right there, and that basically causes everything to fall apart. So the ribosomal subunits dissociate from one another. The bond between the tRNA and the polypeptide chain is broken, so the tRNA is released. And this free polypeptide chain that's been synthesized is now released, folds up, and does what it's going to do in the cell. The release factors dissociate as well. Could these ribosomal subunits then attach to another message and translate it? Sure. Okay. Could these release factors be used over and over again? Sure. Now, molecules in the cell don't last forever. Eventually, molecules in the cell are degraded. I used the word turnover before. And so eventually, the ribosomal subunits will kind of break down. Messages don't last forever. Eventually, messages will be degraded. But could the same message be translated over and over again, potentially? Sure, for a while. Okay. Eventually, tRNAs will be turned over in the cell, so you're going to have to make new ones. But the same tRNA could be recycled and reused over and over again. So, here's a rough summary of the energy demands for protein synthesis. About, and I'll clarify this, I'm going to use the term 3 right now, about 3 ATP/GTP are consumed for every amino acid that is put into a polypeptide chain. 1 to charge up the tRNA, and 2 during each elongation cycle. So that's 3 per amino acid. So a small protein, 120 amino acids in length, and that's pretty small, would cost 360 ATPs to make. And that's equivalent of about 12 glucose molecules going through aerobic cellular respiration. Okay? So it really is energetically demanding for the cell to make proteins. And so the ability to turn on and turn off this process as needed really helps the cell to be more economical energetically. We'll get into that more in talking about regulation of gene expression. I know this is a little hard to wrap your head around the first time you see it. One of the things I will do after the exam on Tuesday is there's some good animation videos that show you this kind of in process and kind of shows you, like DNA replication videos, things happening in kind of quote-unquote real time. So I'll show you some of those, but I would encourage you, if you're having trouble conceptualizing this, to look at YouTube videos, because there's a lot of good ones out there that show this happening. And I think a lot of you probably are more visual learners and respond to that kind of thing. Okay, so for exam Monday, get here as soon as you can, but we do have to stop at 5 till at the very latest. Our format of the exam is the same as it's been before. I think there's a couple less multiple choice questions because we've got one additional question about the video. I will have your video notes up here from the Cancer Warrior. If you have not seen the Cancer Warrior, you better look at it if you want to be able to answer those questions. Natalie has the DVD, and I already gave you out video questions, but I need those given back to me so I can hand them back during the exam. Okay? Have a good weekend.
Sorry.